Hello. So today we are going to be talking about the Krebs cycle, and this is really where cellular respiration begins for us. And it's, in our case, the second step of cellular respiration, but you can think of it as really where cellular respiration actually begins. Because as you remember, glycolysis is just one example of a way that we can break down a molecule into the components we need for cellular respiration, but that's specifically talking about glucose. However, that can happen with any variety of molecules. A couple basics to know about the Krebs cycle. First of all, its nickname is also known as the citric acid cycle. Some people call it TCA for the citric acid cycle. This happens in the mitochondrial matrix. So when we're talking about the mitochondria, actually inside the mitochondria we find the matrix, and that's where this takes place. This Krebs cycle can only occur when oxygen is present, meaning that this is an aerobic process. And so you'll see, once again, after we learn about regular aerobic cellular respiration, we'll talk about anaerobic respiration. And this is where anaerobic respiration has to stop. It can't continue on with the Krebs cycle unless we have our oxygen. As long as there is oxygen present, remember, the pyruvate can enter the mitochondria, go through the intermediate reactions, and become acetyl-CoA. And this is what we need to start the Krebs cycle. The main purpose of the Krebs cycle is to produce NADH and another electron carrier that's very similar called FADH2. And these two substances are high energy electron carriers. Both have been reduced from a previous form. So if you remember, NADH is reduced version of NAD+. Basically, what these molecules do is they move electrons onto the electron transport chain. You can kind of think of them like little drivers that drive electrons to the electron transport chain. And this is our final step in cellular respiration, and that's where oxidative phosphorylation will occur. Also, NADH and FADH2 are very important because in carrying those electrons to the electron transport chain, the ETC is actually what produces most of the ATP. So if you remember, the original purpose of cellular respiration was to break down our food into lots of energy for us. The ETC is where that all really goes down. And without the NADH and the FADH2, we wouldn't be able to have that. So, like I said, NADH and FADH2 are coenzymes that play an important role in energy production. So these guys are high energy, they also carry the electrons, and like I said, these two substances are what allow us to move on to the next step, which is the electron transport chain, which produces the most energy, which is the main point. Here's a little picture to help you remember. So you can think once again of NADH and FADH2 as little guys carrying and shuttling electrons around, like mailmen or truck drivers, something like that. But they're carrying the electrons to the next step of the process. The Krebs cycle, once again, it completes the breakdown of pyruvate to carbon dioxide as well, CO2. So you're going to see that pyruvate, which has several carbons, ends up getting broken down even further and released as carbon dioxide. And then once again, using the pyruvate, it also breaks down this organic fuel through oxidation and generates 1 ATP, 3 NADH, and 1 FADH2 per turn of the cycle. And we'll talk a little bit more about that as well. There are 8 steps in the citric acid cycle, but once again, similar to glycolysis, the AP exam isn't concerned that you've memorized those 8 steps. However, it can be helpful to think of each step and know that it's catalyzed by a specific enzyme. One of the most important steps is the first step, and this is where the acetyl group of the acetyl-CoA joins the cycle by combining with a molecule called oxaloacetate. This combination forms something called citrate, which is an ionized form of citric acid, hence the name citric acid cycle. The citrate is what you need to actually get the rest of the cycle going. And in doing that, there are 7 more steps for a total of 8 that decompose the citrate back into oxaloacetate. And this is why we have a cycle, because once we've gotten the oxaloacetate again, another acetyl-CoA can come in and combine with the oxaloacetate and form citric acid again, once again beginning the cycle. Also worth mentioning once again is the whole purpose of the Krebs cycle is to produce this NADH and FADH2, which essentially shuttle the electrons from the food at the beginning of the process all the way to the electron transport chain. This picture presents a somewhat simplistic view of the Krebs cycle, but I think it's useful because it shows you where everything is moving. So if you remember, when our pyruvate became acetyl-CoA in the initial and intermediate reaction, pyruvate has 3 carbons. Glycolysis, we started with 6 with glucose. Pyruvate each has 3. But one of those carbons got released as carbon dioxide in the intermediate reaction. Then acetyl-CoA, that means acetyl-CoA has 2 carbons. At the beginning of the cycle, acetyl-CoA combines with this 4-carbon molecule called oxaloacetate. When you add those 2 carbon groups together, you get a 6-carbon molecule known as citrate. Once again, this is your citric acid. Then there goes through many more steps where the citric acid is further broken down into another 4-carbon molecule. And in this process releases NADH, here and here, as well as carbon dioxide. FADH2, here's another NADH that gets released. And then in this picture, for our purposes, we mostly are concerned about the ATP being produced. So once again, the ADP in inorganic phosphate goes through substrate level phosphorylation and turns into ATP. But this GDP is a similar molecule that can be found in some animal cells. You don't really need to be concerned with that. I just really liked this picture because of how simple it was. So this is basically what's going on in the Krebs cycle. And as I've mentioned, the most important thing for you to know is what's going in and what's coming out. This is the acetyl group on your acetyl-CoA. This is an even more simplistic version that really just shows you what's going in and what's coming out. And this is from figure 7.10 in your textbook. So we start with our acetyl-CoA. Once again, we have our 2-carbon molecule. It becomes CoA here, and we know that it's now combined and becomes citric acid with the rest of the carbons. 2 carbon dioxides are released. 3 NADHs are released. Our ADP and inorganic phosphate go through substrate level phosphorylation to give us ATP, and we get 1 FADH2 from the reduced FAD. Also, if you take a look at this photo again, remember, because we get back to the original oxaloacetate, this is what allows the cycle to continue. So another acetyl-CoA can come in and the cycle can progress. This is a picture just to remind you that the citric acid is taking place inside the mitochondrial matrix. So glycolysis, remember, does not happen in the mitochondria, but as long as there's oxygen available, we can move our pyruvate into the mitochondria where it will undergo oxidation to become acetyl-CoA, and then that moves on to the citric acid cycle. So what do we get from this whole process? For each acetyl-CoA molecule that goes through the citric acid cycle, we end up with 3 NADH, 1 FADH2, 1 ATP, and 2 carbon dioxide. But remember, blast from the past, after glycolysis, we end up with 2 pyruvates, which through the intermediate reactions are turned into 2 acetyl-CoAs. So since 2 of them go through the process, from the 1 glucose molecule at the beginning of glycolysis, the citric acid cycle actually yields 6 NADHs, 2 FADH2s, 2 ATPs, and 4 CO2s. So you can see this is doubled because we actually have 2 acetyl-CoAs that go through the cycle of the Krebs cycle for each of the 1 glucose molecules we started with. Notice still we have very little ATP being produced. This is going to be important when we move on to the next stage because remember, the whole point of cellular respiration is to harvest the energy from our food. However, glycolysis only gave us 2 ATP, and so does the Krebs cycle. So really, we're not getting a lot of energy from these 2 cycles. One reason for this is because both of these cycles go through substrate level phosphorylation to deliver us our ATP. And if you remember, that process does not yield nearly as much energy as oxidative phosphorylation, which we'll get to in the next section. This is just a summary slide to help you remember the parts of the Krebs cycle that you need to know and be familiar with for the AP test. So the function is for metabolism, breaking down that citric acid, and energy production. But in this case, most of the energy, as I've mentioned several times, comes from the NADH and the FADH2. This whole process takes place in the mitochondrial matrix, so we're now in the mitochondria. This is a simplistic version of what happens in the citric acid cycle. And then the reactants that we need for this are acetyl-CoA, the 4-carbon compound. That's what we really get started with. And our products, we end up with 2 ATP, 4 CO2, 6 NADH, and 2 FADH2. Because if you remember, we actually get 2 acetyl-CoAs from our 1 glucose molecule. So that's why the reaction doubles. That's all for the Krebs cycle, and I'll see you in a couple days for our oxidative phosphorylation lesson.